Hi everyone, welcome to another Coronavirus TV episode. It's Thursday, April the 23rd. Today I'd like to talk a bit about antibodies. We're not talking about antibodies that are in the news in terms of testing whether a patient has had an infection, but rather talking about antibodies as therapeutics. So you may have read or heard a little bit about convalescent plasma. This is, where you take immunity from a patient that's recovered from COVID-19 and provide it into a newly infected patient. And this is being tested at hospitals throughout the world, and it has some success. It's not particularly scalable or viable in large amounts because each donation really only provides enough treatment for one or two newly infected patients. But the logical progression of this is, can we adapt this concept, design our own antibodies from scratch, and then able to manufacture these on much larger scale needed, so that then we can treat, hundreds, thousands, tens of thousands, hundreds of thousands, millions of patients, etc., if needed. And work is being done in this space. If we look at the industry database pharma projects, there are 26 different programs currently in the pipeline against COVID-19 that are monoclonal antibodies. Some of the leaders in this space, Amgen, Regeneron, an interesting new startup called Vir, which has some interesting partnerships. So what the work currently ongoing is optimizing the antibodies. So can we identify and select the best antibody possible? The antibody ideally has high selectivity for coronavirus. Typically, you're recognizing and binding the spike protein that gives the coronavirus its name. You also need to be binding part of the coronavirus that is unlikely to mutate and escape immunity. So the more and more we treat coronavirus, we don't want to be applying too much pressure on it and then creating resistant strains. Antibody also should have good pharmacokinetics and stability. So a single dose would provide circulating antibodies for a decent amount of time, potentially up to a month, if not longer. So at the moment, time is ticking. All of this work is going on, but all the while, the pandemic is evolving and changing. Companies such as Vir and Regeneron are talking about being able to enter clinical trials in a few months, 3 to 5 months, potentially late summer. By this time, the number of new infections, who knows where that's going to be. And I mean, the concern from a clinical trials point of view is that there just simply aren't enough patients with which to test antibody drugs as acute antivirals. Just have to think about remdesivir trials in China, where the number of new infections has dropped so significantly that they were never able to reach full enrollment. In this sense, though, that's actually a fantastic outcome because it means that the other social distancing measures are shutting down the spread of coronavirus to such an extent that we can't test these drugs. So brilliant. The other treatment scenario or setting that these antibodies might find themselves in, if that is the case, is as prophylactics. So if you can get a single dose of your antibody and it provides circulating antibodies for 28 days, this is a form of passive immunity. Patients and people that have received this are unlikely to contract coronavirus if the antibodies are effective. So it's kind of like a vaccine, which is more, the vaccine is the active immunity. In the perfect world, vaccines are developed, the timelines that people are talking about vaccines, if Moderna's vaccine, for example, and other forerunners are successful, then we may have vaccine availability through through next year. So the actual shelf life or useful lifetime of an antibody approach may be somewhat short. The bridge, if these are available towards the end of the year or start of next year, they certainly can act as a bridge to a vaccine, potentially for 6 months or so. You might be looking at giving these antibodies to people that are in key worker roles that are unable to practice social distancing. So you might be talking about as a prophylactic in healthcare workers. So I mean, antibodies sit in this kind of grey area between they don't meet the immediate need for antivirals for the many thousands of patients that are hospitalized with COVID-19 now. They aren't a vaccine either. They will need to be repeat dosed and potentially require intravenous infusion, which is more problematic. So they kind of sit in that grey area. But I think the sentiment around vaccine timelines is certainly shifting, and it is unlikely that the first vaccines will be the best and the most successful vaccines. So antibodies in this sense, as a bridge to vaccines, may be essential in order to have some kind of normality and pharmacological ways of actually slowing the transmission of coronavirus. It may also be that we experience additional waves of acute infections and community-based transmission, in which case an antibody-based drug might be the best at treating hospitalized patients too. So wait and see. The actual kind of useful lifetime of antibodies depends on various other factors that we don't really know about just yet. What should we expect to see next? I expect companies will be disclosing some preclinical data around their select chosen antibodies that they think are the best ones, and then start to get regulatory clearance to enter human clinical trials. I think it's a really interesting topic. Hopefully I've kind of conveyed that, and this is an area that you want to follow too. Thanks a lot for listening, and I hope to catch you soon. Bye now.